demo, what I'll show you is how you can compute chemical shifts from 2D structures of RNA. And so what you're seeing here displayed using var RNA, or three different structural models of one RNA. So this RNA is the same in terms of the sequence, but the secondary structure changes. So these are different uh, conformational states of this RNA. And so for each of these models, what we want to do is compute chemical shifts from them. Now, the reason why we want to compute chemical shifts is that we can use that computed chemical shifts to identify which of these structures is most representative of what you would see in a test tube, right? And the way you do that is by comparing the computed chemical shifts to measured chemical shifts. Another way you can use this is actually say, okay, I have these three structures and I want to attach weights to each one of them, such that when I take the weighted average of the chemical shifts, the computed chemical shifts, they match very closely the experimental reference chemical shifts. And so there are two different kinds of strategies one can take to to leverage computed chemical shifts to be able to model the structure of an RNA. So here these are just again three different states, and so we want to go ahead and compute chemical shifts. So I have saved the secondary structure for each of these models in CT file format. And so what we want to do now is to take these files and utilize them to compute chemical shifts. So my group has worked on methods that allow us to predict chemical shifts from structural models. And so now we have tools for both 2D and 3D chemical shift predictions. And so we have this server, right, this web server, which we call Smalter. Let me just redo this. And so Smalter is just a gateway for structure-based machine learning tools that my group has developed. And one of the tools we have allows us to compute chemical shifts from secondary structure. So for those who are new, you would have to create an account, right? So you can go create an account, and here you add your information, like your username, your password. You have to put in your password again, your email address, and some other information. And you click create, and then I will receive an email. I'll activate your account. Since I already have an account, right, so I'll just go to log in, and then I'll include my username and password information and click here. So once you open up Smalter, you have the first place you get access to is your dashboard, which is you could access also by clicking on this icon here. And what you want to do is scroll down and find this tool called CS-SS, right? So this is a tool that goes from secondary structure to chemical shifts. So you could click on this, right? And then you're presented with this page, and so you have what you call experiments. So you can run an experiment. So you could, let's say I call this CSS as model 1 and give it this name. Default project, we can leave it as default for now. You can always change the project here. Then the application configurations consist of input CT file, an RNA ID, and then a model index. And I'll show you why we need the index in a bit. So let's just take the first of these, right? And we can drag this over here, upload a file, and the RNA ID actually corresponds. Let's just give it any RNA ID. I just use 2L3E. That's the initial PDB ID for the RNA I was looking at. And so because I'm gonna be having a set of chemical shifts for the same RNA but from different models, right, I can actually use an index to keep track of the different models, right? So what I can do is set this index to 1, right? And this is actually going to be used in the output, so that if I combine or concatenate all of the output, then I have an index that I can use to keep track of the different models. So I set this to 1, so I have the initial CT file, the ID, and then the index. And now I can submit this job. So for now, the compute resource that works is actually Bridges, right? But we'll fix it such that Golem also works. But for now, just use Bridges, the default. And now you can go ahead and launch this job, right? And so you're presented with this page, and it gives you a status of the job, right? So here, for example, it tells you the job was submitted. Now it's in the queue. And eventually will be created. So we want to repeat this for all 3, right? And so you could go ahead and click on dashboard and repeat this process, or you can just click clone, right, and actually clone the job that you just ran, right. And so you can now change the name to SS to CS model 2, and you have to delete the old input file, and now you can upload model 2 CT, and here we can actually change the index to 2, right? And then we're gonna hit submit, and let's clone again for the third one. I'm gonna call this model 3, set the index now to 3, and now delete this old CT file and use the new one for CT for model 3, and you submit. So now you could click on experiments, and it tells you can see the three jobs that were ran, that I executed or submitted, and they're all executing. And so these jobs shouldn't take too long. Once they're in the queue, they should be finished pretty quickly. So let's see the first one. Is it still in the queue? It's active. So it's active, so it should be done pretty soon. So we could kind of wait for a bit, and you can see that this job is complete. And what happens is that the output would show up, and here we go. Here we have the output, right? And so you can now click download, right? And so this produces this output. So what I'll do is actually grab this, and I'm going to put it in my test folder. I'll rename it as output 1. Let's go to the other jobs. So they're all completed, so I could go to this job, grab that output, put it over here. I'll call that output 2. So now I have output 3. Okay, let's just expand these. Oh, I didn't want to like that, but anyhow, just to be sure, I'm gonna move this to trash and do this one by one. So first get this one, and this one, and this one. Just so that there's no confusion, I'll just change these names here as well too. You could actually get rid of the zip files if you wanted, right? I'll just put up my trash. Okay, so if you open up these, right, so you can see what's in these files, right, in these folders, right. You have the input CT file, whatever you gave it, and then the output. And so you can take a look at these output files. I'll open them in here, right? And so this is the output, right? And so the first column is actually that index that we specified. And let me actually do it this way so that you can actually see. So I'll do the same thing for this and this. I'll open them up. Would be the edit, right? So you can see if you go from the first file to the second to the third, right? The first column is the index, right? And so that's why it goes from 1 to 2 to 3, right? And again, the reason we want to do something like that is, let's say we want to create one file that stores all of this data, right? We could copy this, put it in here, copy this one, put it in here as well, and then copy this one and put it in here. So now we can store all of these predicted chemical shifts in a single file, and we could keep track of them, right? Because we know that the index is different, right? And very often that's how we utilize these in my group. So then we read this into some using, let's say, to do some analysis in Python. So I could just call this whatever name you want to give it, right? So this file now contains all of the computer chemical shifts. So let's just return to the format, right? So this is the first one is the index, then the residue number in the RNA, then the residue name, and then the atom for which we computed the chemical shift, then the computed chemical shift, and then here you see that RNA ID that we included as well. So this is the data that we have, and we were able to generate this data directly from the 2D structures, right? And so now you have the computer chemical shifts. You can go ahead and do analyses. For example, one of the favorite things we like to do is do Bayesian reweighting of these 3 structures relative to some reference set of chemical shifts. Okay, with that, that's the end of this demo.